Hi, my name is Megan Fastana. I am interested in writing on bacteriophages and understanding the mechanisms associated with host-virus interactions. The purpose of this thesis is to isolate and purify novel bacteriophages, annotate its genome, and analyze host range specificity. Among that, I want to do mutagenesis of the host. Mutagenesis can help identify functions of genes that are important in phage life cycle. I'm interested in this topic because analyzing host range specificity and performing mutagenesis can help provide information on phage therapy. I think phage therapy is an important study for the future, especially with the rise of antibiotic-resistant bacteria. With that rise, the choices for effective treatments using traditional antibiotic compounds is limited. Since bacteriophages infect only prokaryotic cells and not eukaryotic cells, studies into whether using bacteriophages as a tool towards treating these infections is promising. Mutating the host can provide clarity for what role viral genes play in infection. When we know this, we can use our own tools to manipulate the viruses for phage therapy. I want to argue that bacteriophages can be utilized in many ways, specifically phage therapy, but also for drug design and treatments against common biofilm bacteria infections in healthcare. These bacteriophages can be found anywhere, as I discovered mine in Aberdeen, South Dakota. Studying my bacteriophage, moss rose, can provide information that pushes the argument that phage therapy is another valuable therapeutic tool. A possible title for my thesis is Isolation and Bioinformatic Characterization of the Bacteriophage Moss Rose and Mutagenesis of Actinomycete Gordonia rubritincta to Understand Host Virus Relationships. I have listed two primary documents in the worksheet attached to this discussion post. The first link consists of the first undergraduate research journal focused on bacteriophage biology. It includes interviews, excerpts, all conference papers, etc. The second link is a dissertation written by Abdir Majir Al-Harbi. I know I butchered that name. He isolated and characterized his own bacteriophage. The author describes his own experiences with this process. Also, it includes all the items that will be in my honors thesis, such as an abstract, introduction, literature review, etc. These two sources are interesting because I can use them as tools of reference. The first source describes the other's experience with the C-phage program from a different country. It also offers news articles over phage therapy, which I can utilize when talking about applications of my research. Additionally, the second article is a dissertation from a student who isolated his own bacteriophage. His paper could provide clarity for how I should structure mine, as we did similar projects. I could also discover something new that could help me with my experiments by reading his methods. So far, my two secondary sources include a book from the library titled Bacterial and Bacteriophage Genetics. This book mainly discusses various generalized information about bacteriophages and their life cycle. My second source is an article titled Famirator, a bioinformatic tool for comparative bacteriophage genomics. This article provides information on how to use the database Famirator, which shows similarities of homologs in the DR cluster. Moving on, one of my paper's greatest strengths is my data. So far, I have isolated and characterized my own bacteriophage, moss rose. I still have to perform mutagenesis, which is what I will work on this year. But with what I have so far, I think this provides my thesis with a solid foundation. Another strength is the ability to apply my topic to future applications. Bacteriophages have many uses. I know we talked about phage therapy, and doing research on such a popular topic provides me with many sources and pathways to go down. On the other hand, my paper's biggest hurdle will be the findings section. My data relies on good results and well set up labs. I know I discussed how my data is also my biggest strength in my paper, but it can also pose obstacles like contamination, failed experiments, waiting around, etc. Contamination can alter results, which makes it frustrating. I must make sure to use good sterile technique that requires patience, so relying on hoping it turns out can be hard. The good news, though, is that I have time, so if an experiment fails, I will be able to restart fresh. Thank you.